Hey guys, welcome back to the Fields Lab, episode 3. I am a little bit late, but I wanted to bring you something this week about the lab and what we're doing. And this particular talk will be about our grants that focus on cannabinoids and cannabis and how it affects the brain in the context of inflammation and immunometabolism, right? Because everything we do in our lab, pretty much everything we do in our lab has comes from that perspective of how HIV or Alzheimer's affect immunometabolism, and in this case, how using cannabis may exacerbate these issues or prevent these issues. And so we'll be talking about people that use cannabis, the interactions with people who have HIV, a little bit of interactions with people who may be developing Alzheimer's disease and aging, and the endocannabinoid system, the system which cannabinoids bind, that they sort of hijack in our body. The reason that they are effective to us is because we have an endocannabinoid system and that's the system that they interact with. Um, and then maybe possible therapeutic uses. And then I'll go into our grants and how we are looking at the endocannabinoid system in the context of immunometabolism of the brain and personalized medicine. So basically, how cannabinoids may affect different people, how we may identify the people that cannabinoids for whom cannabinoids may be beneficial versus the people who maybe they should stay away from it. And I am not endorsing the use of cannabis at all, but it is used medicinally, and there is some. I'm not endorsing the use of it recreationally, just to be clear, and I'll explain why. But there is some evidence that in disease states, cannabis may be beneficial. So that's kind of how we'll go. I put together a little PowerPoint just to kind of outline my talk and what I want to talk about. Keep me on track because that is not an easy thing for me to do. I love going off on tangents. And then I just wanted to say this work that I'll talk about is only possible because of a bunch of people I work with, a bunch of clinicians and statisticians and psychiatrists and psychologists and virologists and all kinds of folks that actually work with the people. And then my lab, where we actually take the samples and we analyze them. And we've got people doing brain imaging like MRIs and PET scans. And in the future, I'll explain some of the things that we have found, some of the discoveries we've made. And yeah, I'll show you. We already have a few publications on these grants, but more will come. So let me go through this process of sharing, which always seems to be a sticking point for me. All right, so here is a little PowerPoint. I'll make it a little bigger, probably make it a little nicer. Um, Continue to share. Yeah, so cannabis in the brain. Again, when I was looking, I wanted to make sure I wasn't being super redundant, which with what is out there on YouTube and just on the internet in general. There's a lot about how marijuana and cannabinoids affect neuronal function in the brain and like memory and reward systems, anxiety, and that is all pretty well documented. What I didn't find as much of is how cannabis can affect inflammation in the brain and how the immune system interacts with the brain. And I mean, some people would say the immune system is, you can't hardly study the brain without considering the immune system, particularly brain diseases. And I'll probably have to go a little bit, I'll touch on that a little bit, but I'll go more into that in future episodes. But yeah, cannabis in the brain, a little bit. I'm no expert on the endocannabinoid system. I'm much more I've I've spent a lot more time, have much more experience uh, investigating the immune system in the brain and brain cells, which I've explained before. But we're getting into the cannabis, the endocannabinoid system, and how it interacts with the brain. So I will, in the future, I think I'm going to bring other people on to explain the my weak points, and hopefully I won't mess them up too bad here. So cannabis, the highlights. I just thought kind of give an overview um, of why people are looking at cannabis. For one thing, it's been used by humans for thousands of years. Uh, it's the most used illicit psychoactive drug in the United States for sure, with 61 million people having reported using it in the last year. So that's almost, yeah, 20%. Well, not every five people. So a lot of people use it, some recreational, some uh, medicinally, and I'm not sure if that's all people that are using THC. Some people just take CBD. They say it has a calming effect, but it's not like psychoactive. You don't get real high with it or anything, but THC is the one that gets you high. And THC and CBD are the famous cannabinoids, the ones that everybody has heard about. And let me back up and just say it probably THC and CBD and every cannabinoid can affect almost every organ in the body because, as you see in this picture here that I borrowed from Kalapa Clinic, you can see their website there. So I'm citing them. Um, the endocannabinoid system is present and active in pretty much every part of the body. Um, we know we've discovered a couple receptors, not us, but the scientific community, CB1, cannabinoid receptor 1, and cannabinoid receptor 2. And generally speaking, cannabinoid receptor 1 is expressed on neurons in the brain and the peripheral, so outside the brain, outside the central nervous system. Cannabinoid receptor 2, they would say, is more in immune cells, but I can tell you it's not that clear cut. And sorry about that. There are likely receptors other than CB1 and CB2 that are involved. And I don't want to get too technical, but I just want you to know there's lots that we don't know. Cannabinoids, they don't, I guess I should talk about receptors, and I'll talk about the endocannabinoid system in a second. But let me just say that they're in the plant, there's THC and CBD that bind these receptors, but there's also, I think I've heard well into the hundreds of other cannabinoids that have been identified in the cannabis plant, and they interact with the endocannabinoid system. And the endocannabinoid system, it describes basically for THC and CBD. I think the important thing to know is that we have these receptors on our cells that we produce naturally so that the endocannabinoid system can produce endocannabinoids, and they can be released from some cells. They bind to receptors on other cells to communicate. And what is known, or what is sort of the consensus right now, is that it's used as to restore homeostasis in times of stress. And I'm talking about immunological stress, like if you have an infection, possibly if you have depression or anxiety, or maybe heat or cold. I'm sort of speculating there, but the point is, whenever your body gets pushed in one direction away from, and it gets stressed, the endocannabinoid system, it kicks in to restore things back to homeostasis, or a functional sort of your physiological status quo, so to speak. And in our lab, we found that is probably true, or at least when we look at the endocannabinoid system in the brain during disease, it's upregulated generally. And that's probably to try to restore physiological homeostasis. And it interacts with all these organs. So whenever you take THC or CBD, or they have CBN or CBG, and then there's a whole bunch of others, it's probably affecting a lot of different organs. You just feel it in the brain. But again, we're going to focus on how it affects the immune system. And this kind of already talked about this, but the endocannabinoid system, there's endocannabinoids, 2-AG and anandamide. These are just two molecules that we produce. Lots of animals produce them. I'm not sure how far back it goes, evolutionarily speaking, uh, but for sure, mammals, we all have the endocannabinoid system. We have the receptors so that these molecules can bind and communicate to cells, the same receptors that THC and CBD bind. And then we have the enzymes that produce the endocannabinoids, enzymes that break down the endocannabinoids, and it plays a role in a whole bunch of behaviors. And just I listed what I found here, but sleep, mood, appetite, reproduction, stress, nerve function, inflammation, and pain. We really focus on nerve function and inflammation, really more the inflammation and how that affects nerve function. But in some cases, we have studies on mood as well because we look at depression and how people using cannabis may have different rates of depression. I'll go into that later, probably in another episode, because I don't think I can get into all that here. So there's a big concept that I kind of have to explain, and it's something that, even though you probably haven't heard this word a whole lot, it is prominent in our society today. It's called hormesis, and it's basically the idea that a certain amount of stress is good for you. And the small amount, and you kind of titrate the stress. You don't want too much of it, and as it goes up, it gets to where maybe it's not as good for you, and then once you have too much of it, it starts being bad for you. So it's like this. It's not linear, as in it doesn't keep going in the same direction. This graph kind of shows it. As you have the amount of the stressor. Here we go. I couldn't find my cursor. As you increase the amount on the x-axis, you'll see the benefit increases, but as you keep on putting on more and more of the stressor, all of a sudden you start getting diminishing returns until actually it's harmful. Examples would be like caloric restriction. We obviously need food, but there's good evidence that if you don't eat all the time, you don't always need to be stuffed. And things like intermittent fasting, or there's several different kinds of fasting regimens, but there's good evidence that if you don't eat all the time and you allow your body to go without food, it actually recycles the damaged parts of cells, like damaged proteins, damaged organelles. They get recycled first and it clears them out. And then you start eating again. You don't want to stop eating forever, right? Then you end up dying, right? Of starvation. But some stress by way of caloric restriction is good for you. Hit workout. So that's the idea. If you ran a marathon every day, probably not so good for everybody, although I have heard of somebody doing it for like a year. That was an extreme case. But most people, you work out a little bit, you stop, you recover, and you work out a little bit more. These high intensity interval workouts can be good for you. Going from hot to cold. I actually sat in a cold plunge this morning and a sauna. You don't want to sit in a cold plunge for too long, you get hypothermic. But if you do it for like 5 minutes, there's a lot of evidence that it's good. And then the same with the sauna. You can go sit in a 180-degree sauna for a little while, but you don't want to sit there too long. Same thing with cannabis. This is the idea, or at least, yeah, there's some evidence that a small amount of cannabis could be good for people in disease states, like people that have possibly dementia or HIV, for example, HIV-associated neurocognitive disorders. But using it a lot, it may not be so great. And there's evidence from our group, studies in the people I work that have shown this. There's evidence in mouse models of Alzheimer's and dementia that if you give low doses of THC or CBD, you can improve their outcomes. And how this translates to humans, and for example, I'll talk about that in a second, but with people with HIV, there is some evidence that people with HIV that use low amounts of cannabis have less inflammation and less cognitive impairment. But if they use more cannabis, it's those benefits go away compared to people with HIV that use no cannabis. And now we can compare it to people without HIV that have the same age matched control, so they're around the same age, using the same amount of cannabis, and you don't see the same types of benefits. Of course, they shouldn't have their controls, so they shouldn't have the cognitive impairment anyway. So this hormesis may be beneficial for things like pain, people with cancer that can't eat, so it increases their appetite. There's some evidence for anxiety. We'll talk about inflammation and neurodegenerative diseases. So here we have two studies. We have two studies that are like really focused on trying to figure out how different cannabis use patterns affect inflammation and how that affects the brain. And these are actually schematics from each of the studies that we had funded. This first one on the left side, I think we started in 2020. And so it's been going a while. We've got a few publications out and several that are in the works. And then the one on the right is actually looking at cannabis use and the effects on the blood-brain barrier, which I don't want to talk too much about because it gets kind of complicated. But basically, your brain, just like every other part of your body, has blood vessels running all through it. And these blood vessels, your brain has to be protected. You can't just let everything that runs throughout the rest of your body get into your brain, into your neurons. So the blood-brain barrier actually is an extra layer of protection that wraps the blood vessels to keep it's like a gatekeeper for what gets inside of your brain and interacts with the neurons and the microglia and the astrocytes, all those cells that we talked about before that need to be protected in the brain because the brain's pretty important for controlling the rest of your body. Well, it turns out in almost every neurodegenerative disease, probably everyone, I just haven't studied everyone, the blood-brain barrier is compromised. And there's evidence that HIV definitely compromises the blood-brain barrier, and cannabis may keep that from happening. So I already told you that there's some evidence that low levels of cannabis could be beneficial. Low levels of cannabis use could be beneficial for people with HIV and with Alzheimer's disease. For example, the first study, I'll go back to it now. It's trying to investigate how these different levels of cannabis use affect inflammation. And what we've done, and these are people I work with, both these studies are with a big group of people. I'm just doing one cellular aspect of it. So we recruit people with and without HIV, and we split them up by how often they're using cannabis. And we confirm that by measuring the THC metabolites in their urine. And then we also take their blood and we measure a bunch of inflammatory markers and we measure components of the endocannabinoid system in their blood. And then in our lab, we take the blood and we actually isolate these macrophages, which are immune cells that are kind of like the police officers. I may have talked about them before, but they're kind of like the police officers of the body. They run around, they float around your circulatory system, and whenever they see a problem, they crawl out of the blood vessels into the tissue and they start looking around. They have all these receptors and they're trying to figure out what's going on. Well, they also have cannabinoid receptors on them and they also go into the brain. So in this study, what we're doing is we have, I think it's 60 people with HIV, 60 people without HIV. And then we're trying to get about 20 uh, from each of those groups. We want 20 that don't use cannabis at all, 20 that use it, what we'd say is low to moderate use to model the low amounts of cannabis use, and 20 that use it a lot. And our hypothesis is we think that the people with HIV that use low amounts of cannabis, they'll have less inflammatory markers on these macrophages. They will be less immune reactive. Um, which would translate to less neuroinflammation, and it would protect neurocognitive function, which is why we think that people with HIV who use low amounts of cannabis may be protected. And we think that this will not be the case in the people without HIV. And we've got some evidence that that is true. And I can present that paper later if y'all are interested, but I'll probably get somebody else on here to talk about it with me, um, because there's other evidence as well. But we think that that may be true for some people with HIV. And this is where we get into the personalized medicine. It's pretty clear that everybody with HIV does not react the same way, right? They end up you end up seeing that some people cannabis may be anti-inflammatory in them, and some people it may not be as much. And that's okay because doing these types of studies, we think we can pick out the people that we hope that we can use these sort of assays to pick out the people that may be amenable to treatment with certain cannabinoids, and then other people that maybe they should stay away from it. In the next study on this right schematic, I should also say in both these studies, we're also doing brain imaging. So we're doing taking their blood, we're measuring their inflammation, their endocannabinoids, measuring their cognitive status, their depression status, and then we're putting them in an MRI to try to identify if they have more inflammation, which parts of their brain is functioning, how well it's functioning. And then we can correlate all these things together. And then in our lab, we're taking the cells to try to find the mechanisms that may be causing these changes, and if cannabinoids are underlying these changes. The second grant is very similar, but we have a focus. It's got a similar layout, but we have a focus on uh, the blood-brain barrier. And so the blood-brain barrier, it is basically the same blood vessels that run through the rest of the body, but the cells, so your blood vessels, you can see here, I hope you can see my cursor. It's basically endothelial cells, a single layer of endothelial cells. In the blood-brain barrier, though, there are pericytes that wrap around them, and then astrocytes, which I've probably told you before, my favorite type of cell in the brain. They wrap around the pericytes and the endothelial cells. So you have multiple layers wrapping around the blood vessel, and you can have perivascular macrophages, which are the cells that we are investigating. One version of the macrophage wrapped, and they'll be circulating around the blood-brain barrier as well. And there's good evidence that in neurodegenerative diseases like HIV, Alzheimer's disease, this blood-brain barrier is compromised. And what that means is the astrocytes, there's like where this is sealed, there's gaps because the astrocytes and the endothelial cells and possibly the pericytes, they react to the inflammation and they allow more immune cells to get into the brain. And so, and we believe, and there's good evidence that there is a mitochondrial component to this or a metabolic component, that the HIV is altering the metabolism, which is interconnected with the immune reaction, and cannabis may be muting this, or certain cannabinoids may be muting this. So again, we are taking the blood and we are culturing the macrophages from the blood, and then we are taking the media, the juice that the food that they grow in, and we're adding that to endothelial cells and astrocytes to see how it affects their mitochondrial function and their inflammatory reaction, basically their inflammatory products. And then just like the other study, we are correlating that with behavioral assays, how much cannabis they use, how many cannabis metabolites we find in their blood, their endocannabinoid system. And hopefully, and we think we already have evidence that we are able to see that how different levels of cannabis use affects the brain function in this way. And again, we think we'll find people and maybe even certain cannabinoids that help some people. Maybe some people shouldn't use it. Maybe it's more beneficial in disease states. Maybe for some people it's beneficial anyway, even if they don't have HIV or some cognitive impairment, or maybe it's really damaging to them. Those are the kinds of things that we think that we can identify with these sort of projects and assays and the data that we will generate. I'll also say that we are looking at macrophages, but there are many other immune cells in the blood, natural killer cells, T cells, and cannabinoids apparently affect how they function as well. It's We are not going to be able to look at that because all these assays take a lot of time, and so you just need more people and you need more reagents, and everything has to be expanded. Um, so this is all we're looking at now, but we will try to look at other cells in the future. And what's cool about these grants is these projects is we're not only are we looking at the immune system, we're also using the imaging and the behavioral assessments to see how the neurons are reacting to these different levels of cannabis use, and the mitochondria, how that all feeds into it and how they change. I think that's probably all I'll say right now. I don't think I have another slide. No, that's it. But hope you like this, and I'll be back more next week. Thanks.